If you have a basic understanding of what DNA is and what it does at the most superficial level, then you've got enough background to understand the rest of this talk. My name is Aaron Kimball, and I'll be speaking about the data-driven future of biotechnology, how big data and the Internet of Things are changing how we approach bioengineering and are fundamentally changing what we can accomplish in this technological sector. To start out, to give you a little bit of background about myself, as said, my name is Aaron Kimball, and my career goal has been to find interesting problems where big data can lend an advantage and build solutions in this space. I've been working in the software industry for about 7 years. I started out as Cloud Aaron, got my hands dirty with Hadoop, built the Apache Scoop project. I then worked at a company that I founded called Weeby Data, where we built big data applications on top of Hadoop and HBase. And for the past year and a half, I've been working as the CTO of Zymergen. Zymergen is a software and data company that's revolutionizing bioengineering with a combination of machine learning, automation, and data-driven process improvement and search techniques. And we use these capabilities to optimize the genomes of microbes for chemical production. I will unpack that statement for you in a couple more slides. But first, just to give you a quick overview of how the rest of this talk is going to go, I'd like to start out by giving you some background information on industrial fermentation. This is the sector in which Zymergen operates. And then talk a bit about DNA optimization, which is the role that we play in the industrial fermentation sector. Then we'll go deeper into the technology side of this, talk about the architecture of our software system, and touch on, in even greater detail, a few key components that I'd like to highlight for you today. So let's begin by talking about biology, about industrial fermentation. What is industrial fermentation? It's very similar to the kind of fermentation that should come readily to mind when you hear that word. In regular fermentation, you put yeast in a vat, you feed it some sugar, wait some time, and you get delicious beverages out the other side. In industrial fermentation, by and large, the same process happens. You put yeast or bacteria or other microbes in a large fermentation vessel, feed it sugar, wait some time, and get something probably less potable out the other side. Industrial fermentation can be used to create a whole number of different compounds, from commodity and specialty chemicals to agricultural products, therapeutic drugs, artificial flavors and fragrances, and a whole lot more. Industrial fermentation is one example of a broader technological capability called bioengineering. In bioengineering, you change the DNA of organisms in order to achieve a beneficial effect. So industrial fermentation focuses on changing the DNA of microbes to turn them into tiny factories to make chemicals for us. This can be used for biofuel, for example, creating high-proof alcohol ferments or creating other chemicals that can be burned in place of traditional petroleum-based fuels. We can also use this technology to take advantage of nature's existing capability to create chemicals. All sorts of microbes emit effectively as waste products a huge number of chemical compounds that we find useful. For example, amino acids are often used in fertilizers or animal feed enrichening. But for every 100 grams of sugar that is fed to a collection of microbes, perhaps 1% or even 0.1% of this turns into that amino acid that you're interested in. By changing the DNA of the microbe, this can be increased to become 40, 50, or even 60% of the output of the metabolism of this organism. But we're not restricted to just the chemicals that nature would produce all on its own. We can refactor the metabolic pathway, which is to say, we can create a new route of digestion through the organism so that it takes sugar in and converts it through different chemical processes into output chemicals that it never would have made in the first place, such as industrial chemicals like solvents, thinners, adhesives, lubricants, and more. Bioengineering also has applications in the medical sector. Some of these are based on industrial fermentation. Therapeutic drugs such as penicillin are industrially fermented products. But we can also use bioengineering for other concepts like gene therapy. Gene therapy is a new kind of treatment that's used to help people with certain genetic illnesses. A virus that has been inactivated and filled with a custom DNA payload is injected into the patients, where this DNA helps repair the genes of the affected individual and helps make them healthier. Bioengineering also has applications to biological computation. Like software engineers, biologists frequently use models or abstractions to reason about what they're working on. And one of these models is taken from the electrical engineering sector, a circuitry-based model of the cell. And this circuitry-based model can be used to create biological sensors for harmful agents that might be in the atmosphere in our environment and create early warning systems or other kinds of detectors. All of these applications are becoming possible because of the increasing routineness of DNA construction. This chart shows an exponential logarithmic axis on the y-axis and time on the x-axis. The blue line shows Moore's law, the doubling every 18 or so months of the transistor density and reflecting a drop in cost of silicon-based circuits. The red curve shows our productivity at reading or sequencing DNA. You can see that for the past few years, we've made extraordinary gains in this sector, and it's actually outstripped Moore's law in its speed. The yellow line, which is lower down, but still increasing at an exponential rate, is our productivity at writing DNA, fabricating custom DNA sequences for our particular purposes. But while we can construct any given DNA string relatively inexpensively, optimizing DNA, choosing the best DNA sequence to perform a particular function, remains a very hard problem. In order to make changes in a DNA sequence, you're effectively reading the machine code of biology, and our models are not sophisticated enough for us to fully understand what is happening at the nucleotide level at all positions. So to get DNA optimization right, you need to do two things. First, you need to model DNA more effectively than human intuition is currently capable of doing. And second, you need to test changes a lot more efficiently. And by testing efficiently, what I mean is performing these operations at high throughput and using high precision and accuracy when taking measurements on your DNA samples and the microbes that they're in. So this DNA optimization space is what Zymergen does. We optimize microbes for chemical production. Our optimization workflow has 4 basic steps. The first step in this cycle is to design the new microbes in the computer or on paper, so to speak, to come up with an array of changes from our best current strain of the microbe. Each change might be in one position, and we have 100 or 1,000 variants at a time that we want to test. We cannot do so-called in silico testing. We can't simulate what these DNA changes are going to do at that micro level and know whether it's going to raise or lower the productivity of the microbe or simply do nothing. So we physically construct microbes that have this new altered DNA sequence in it. We then put these through their paces in small fermentation vessels. These microbes are fed sugar, and their productivity at creating the chemical we care about is evaluated. In the final analysis phase, we incorporate data from all 3 of the previous aspects of the cycle and incorporate these test results into the next design phase. If a couple of the DNA permutations showed a beneficial effect, those changes might get aggregated into a new leading strain, which is used to generate 100 or 1,000 more candidates for the next cycle of engineering. As I said, we have to physically build all of the strains that we want to test. And we want to test hundreds or thousands of strains at a time. And that means an awful lot of work with a tool like the one shown on the left side of the slide here, a pipette. Our wet lab uses a lot of pipette work. And if you have to build 100 or 1,000 strains, you might decide that this is too tedious an instrument to use, so you might look for some parallelism. This 8-channel multi-pipetter, for instance, can help you move liquids or DNA samples from 8 input test tubes to 8 output test tubes at a time. If that's not enough parallelism for you, you could look to this 12-channel multi-pipetter, or to bring out the big guns, a 48-channel multi-pipetter. But while raising the parallelism of hand tools has been a proven business model in certain industries, this will not scale fast enough for us to accomplish what we need to do. Instead, we've turned to robot automation. Automation raises the bar in a fundamentally new way as to what we can achieve in terms of throughput, consistency, accuracy, and speed. We can move hundreds of samples of liquid at a time in a much shorter amount of time than a human being careful in a wet lab would be able to accomplish themselves. We actually refer to our wet lab as a factory because we have automated every step of the process that we perform. We use robots from start to finish. But with increased automation, come increased analytic burdens. Now that we can construct hundreds of thousands of strains at a time, we have to come up with genomes to test. We also have to track workflows that are moving through this laboratory at high throughput. And when all of your workflows involve hundreds of samples of colorless liquids, it's easy to get things confused if you're trying to track this manually. And finally, we have to correlate the results of all of our assays or studies with the genomes that we generated in the first place and use this in a feedback loop. Wet lab management software, by and large, has been designed with the understanding that it's being used by humans performing wet lab work manually. High throughput automation requires a fundamentally different approach to laboratory management software, which we are taking in as we design our software stack at Zymergen. The improvement loop that I showed you in a few slides ago with respect to how we improve DNA is a specific case example of a broader concept, a concept of a perpetual quality improvement loop. Every time you run a cyclic process, you get more and better data on processes and results. This, through analysis, can lead to better predictions about how future cycles of engineering will work, better understanding of where failures occurred and why, and allows you to perform data-driven workload determination and optimization. This gives you greater confidence in the methods that you're using to perform the work and optimizes the running time of your cyclic workflow, improving the throughput and overall leading to more and better data on processes and results. I believe that the lessons that we can take out of this and my case study here can be applied to many of the industries that you work in as well. Naturally, we couldn't do this work purely by hand. We turned to a lot of software components in order to enable this sort of cycle to achieve the harmonious forward flow that we're looking for. So now that I've set the backdrop for the context of how our work gets done, I'd like to get into more detail about the particular software that we use to accomplish it. The factory robots that we use are general-purpose laboratory robots. They have pipetting tips and other instruments that can be used for a variety of different protocols and can be reconfigured through programming. Between these reconfigurable robots and the software stack that we have built, we have effectively delivered a software-defined factory. Several different optimization workflows that are particular to different chemicals or different microbes can be employed in the same facility and redeployed on a week-to-week basis. Helix is Zymergen's in-house software for DNA design and analysis, and it plays a role in every step of our optimization workflow. Some specific functions that Helix performs. First, in the design phase, we use the software to design optimized genomes. We then use the software to track the genomes through the build and test phases of factory workflows. Quality control analyses happen at several steps of the way to ensure that we're building what we believe we're building and put error bars on our other test results. We need to integrate data from multiple sources across this workflow and then use all of this information to calculate microbe performance relative to the previous generation. Are our microbes getting more efficient? This slide builds out to show the data and system architecture that we've built at Zymergen. The data ingest in the core of our system happens through a collection of REST APIs. These REST APIs validate all of the data that comes in and store it in different back-end data stores according to the kind of information that we have. Data is then replicated out to a number of secondary storage tiers where the data gets transformed physically into different shapes that are particular to different kinds of analysis. All of these systems are then used to feed our various front-end tiers. The Laboratory Information Management System, or LIMS front-end, is used by our research scientist and our factory operations team to upload or inspect virtually every piece of information in the system. Our factory operations team also leans on workflow tools to help guide their work from start to finish, tracking which plates of material are in which phases of the engineering cycle and what needs to happen next. Our automation systems bridge the gap between our REST APIs and our data sources and the robotics themselves, providing information and directives to the robots as to what they need to do next, and also taking measurements that were captured from the robots and the instruments and uploading those into our back end. Finally, our big data processing systems are used by our data scientists. So that's the highest level architecture of the system, and I'd now like to go into more specific detail about particular elements of our software stack. So let's walk through the cycle of strain engineering and see how it affects it at each point, starting with the design phase. For our scientists to design new genomes, they turn to a custom genome design language that we've developed called codon. The snippet of codon shown on this slide will take a host strain of E. coli and modify it by loading a collection of short snippets called promoters into the system and inserting those at various points throughout the input genome. I'll go through this example in more detail and with a little more background in just a few more slides. But first, I'd like to take a step back and talk about the high-level goals of the language. First, it needs to be useful for high and low-level work on sets of genomes. By high-level work, I mean operating on large blocks of DNA at a time, removing or inserting whole genes or other components. Low-level work works at the sort of machine code or assembly language level of manipulating individual nucleotides or small nucleotide snippets. Also, remember that our lab works at high throughput, so it would be inefficient for our scientists to spell out every individual change they want to make. Our operators need to work on sets of genomes at a time. The output of a codon system needs to be a set of data structures we can use to schedule our factory work. It needs to have an easy learning curve for scientists. Many of our scientists know SQL. A few of them know Python. So getting them to use a programming-based system, we need to make this very approachable for them. And so it is very important to us that our scripts be concise and express their meaning in only a few lines. We also want our scripts to always terminate. We don't want to have to roll out upgrades by upgrading software that's deployed on individual scientist laptops all the time. So we want to run their scripts for them in the back end, but we don't want them to give us runaway infinite loops. And finally, these scripts need to be transactional. We're going to generate a collection of data structures that capture a campaign of work to put into our factory. And if the script has an error, we don't want to upload half a campaign and clutter up our data sets. So we implemented this system as a custom DSL written in Scala. And the genomes and the edits to these genomes are described as a collection of Avro data structures, which I will again go into more detail in a few more slides. The codon system is hooked into the rest of our REST APIs. So we use the Jersey REST client to allow scientists to load DNA strings and sequences and other metadata from our live system. And then this also pushes the work orders back through this REST API when they're ready to place an order. We turn to other open source tools like BioJava for manipulating DNA sequences and reading and writing file formats that are common in computational biology work. And finally, our users submit their scripts through a web-based form so they don't need to install and configure a programming system on their own computers. So I'd like to tell you a bit more about that short script that I showed at the beginning of this section. But first, I need to give you a bit more background on the logical model associated with DNA. Here is a random DNA sequence. And so we can see that you can read it like a string, a collection of the nucleotides A, C, G, and T, moving from left to right. But what we can also see is that sections of this are colored. They're blocked out as annotated elements. Each annotation can be read as a string at this high level as well. So on the low level, you can see a promoter called P1 and a gene called YFG1. And so we could view a larger genome in a more abstract form as P1 followed by YFG1 followed by a number of other elements. So each of these annotation data structures has a type associated with it, a gene or a promoter or some other functional element within the DNA. And it has a name. This name is a bit of a science in-joke, by the way. It's the foo or bar of science. Scientists love abbreviations, and YFG is your favorite gene. Annotations also have properties associated with them, and this is a free-form map of keys and values. Some of these properties are common, the author who put the annotation in place, or synonyms, maybe this is YFP. Sometimes elements also have properties that are particular to the kind of annotation that it is. So this promoter is associated with the terminator T1 that follows the gene. Our genome editing functions can operate on DNA as if it's just a string of letters, functions like concatenate, delete, replace, insert, and so forth. But we also have genome-aware edit operations that operate on the annotated DNA level. The replace promoter function, for example, replaces the nearest promoter attached to an annotated region, or inserts the promoter if that region was not previously driven by a promoter. So this is an example of one of the kinds of optimizations that we can perform on genomes, and it's called promoter swapping. A genome obviously is a sequence of genes, and genes are what provide the behaviors of an organism or its traits. But a genome is not just made of genes. There's many other kinds of functional elements inside of a genome. Each gene gets preceded by something called a promoter, and it gets followed by something called a terminator. A promoter is like an amplifier or a volume knob for the gene that follows it. It determines how hard the effect of that gene will be manifested in the organism. So by raising or lowering the volume levels associated with different traits of these microorganisms that are indicated in the metabolism from sugar to the chemical that we care about, this allows us to change its behavior and hopefully raise the production of that chemical. So what we might want to do is load a library or collection of these promoters, say P1' through P3', and try them in front of each of YFG1 and YFG2. So the codon statement shown at the bottom of this slide will find all the annotations that match the pattern YFG star, and it will then replace the promoters associated with these genes one at a time, generating 6 output genomes where each of P1 through P3 is shown in front of YFG1 and YFG2 once. So this is one example of the kind of mutation that we can load into our data model. As I mentioned, a codon script executing a function like replace promoter is going to generate data structures describing the changes. And we store these data structures using Avro. The main rationale we chose for this is strong support for nested records and also free-form maps of keys and values. We also have prior experience working with Avro. We store the writer schema for each object next to the blob for the object so that we can continue to reconstitute these even as we evolve our schemas over time. Each individual object in our system is immutable. We write it once and persist it, and then we reread it many times. As our factory system transforms the understanding of the work order, we generate new data structures representing each phase in this chain. There are 2 primary data types in this data model. One of these is called a DNA component, which stores a DNA sequence and its associated annotations. And the other is a DNA specification, which describes an operation on this DNA. We designed these data structures specifically to be compatible with other common file formats or exchange formats used in computational biology so that we can interoperate easily with the other tools that our scientists use. I'd finally like to make sure that I give credit to my colleague Barbara Frowen, who's the one who designed these data structures. So it's really her work that I'm presenting on these next few slides. Let's start by talking about DNA component. A DNA component stores a DNA sequence. This could be directly encoded as a string, but for space efficiency reasons, we might also simply record a diff between this and some other prior genome strain. The DNA component holds common metadata like the name of the genome, its description, the source if we loaded this in from literature, and it's identified by a universally unique ID within our system. We also store a collection of the annotations in the genome. Each annotation has an extent, a start, and a finish offset, as well as a name and a type. The types can't be just anything. The types are actually validated against something called the sequence ontology, which is a collection of types of functional elements inside a genome that's been developed by a consortium of biologists and open sourced. The sequence ontology also dictates which additional properties might need to be associated with each element of the genome. We finally can store additional arbitrary properties at the top level of a DNA component. For example, the ID numbers of similar DNA sequences or aliases for this particular sequence. The DNA specification describes an operation on sets of DNA operands. And these operations all occur at the low string level. So a function like replace promoter gets resolved down into functions like replace, subsequence, concatenate, delete, etc. And all of these are binary functions. And so concatenate, you might think, takes 2 strings and sticks them together. All of our binary functions actually take lists of inputs, a list of inputs for the left argument and for the right argument. These lists can be combined with either a dot product, where we sequentially co-iterate over these elements, or a cross product where we generate a matrix of outputs depending on the preference of the scientist who's developing the campaign. DNA specification also includes additional string-based arguments to some functions, as well as properties of the overall work order. For example, the suggested lab protocol to use to physically affect this operation, or metadata templates to attach to the DNA components that would represent the outputs of this function. DNA specifications are also identified by a common, unique, universal ID. And these functions can be recursive. The arguments are actually ID numbers on both sides. So these can be the ID numbers of direct DNA components to operate on, or they can reference other DNA specifications, creating chained orders that carry a complex collection of edits to a genome. So a DNA specification gets placed as a work order into our factory. So let's see what happens with our software as we move into the factory. When tracking a factory, we've had a few major goals that we've tried to keep mindful of. First, one of our main responsibilities is providing validated key metadata across all factory operations. Data quality is of paramount importance in our system. At the same time, we need to make our data types extensible by our users, because the engineering team would have no hope of keeping up with the complexity of the data model if they had to add individual data elements manually ourselves. We've also created a unified namespace or ID number space for all our factory data records. This allows us to share extensions to our data model commonly across all of our individual record types. For example, quality data can be applied to strains, samples, plates, wells, and many other types of records with no specific integration work on our part. All of the information that we store in our system is accessible to our users through our LIMS. Here's a screenshot that we take from it showing a particular experiment. At the top of the slide, there's a large amount of metadata about the experiment itself. When did it start? Who's the username who created it, and so forth. And this experiment is also linked to a number of plates of material, each of which has their own metadata that we track. Not shown on the screen here are all of the results of measurement systems applied to these plates within the context of this experiment. Our data models were designed with extensibility as one of the paramount design principles. Everything exists in a unified key space. We call these Zymergen IDs, or ZIDs for short. We also track, along with each record, the serialization information necessary to reconstitute it, be this the type of record, an Avro schema that allows us to deserialize it, or so on. Our core metadata is all validated by our REST API using business logic that's appropriate to each particular data model. Some of this information is common to all of our models. For example, the ZID number, the name of the object, the username who created the object, and so on. And some of it is obviously particular to the data model. So a plate tracks the number of wells contained within that plate. We also have extended attributes, user-added, user-defined key value pairs. This data is also typed, so it can be validated in that regard. But the namespace of keys is developed by a small collection of users outside of the software engineering team. We limit that aspect to a small collection of users so that they can keep most of the data dictionary hopefully in their heads and reduce the duplication of different keys that might mean the same thing. But we've provided them with lightweight interfaces through our LIMS front end to extend the collection of valid keys. So as a particular example, let's talk about plates. Plates are the basic unit of high throughput laboratory manipulation. Physical individual test tubes are too bulky to use at high throughput. We would need to have like millions of them. So instead we use plates, which are plastic containers of wells, which are typically 96 to the plate. They sometimes come in 384 to the plate. And each well can hold perhaps 5 or 10 milliliters of liquid. And for each plate, we track a number of different metadata elements. Obviously, a unique ZID, the name and description of the plate, the project that it's associated with, the particular kind of plate hardware that this represents, the number of wells, and links to the upstream and downstream plates in our workflow. Plates are to barcodes as peanut butter is to jelly. They're always stuck together. Every plate in our entire factory has a barcode on it. We actually have a robot whose job it is to put barcode labels on the sides of every plate. So here's an example barcode from our system. It contains the ZID number in both 128 barcode format and a human readable format, and I'm told it's a human readable plate description in the lower right, although opinions vary on that subject. These barcode labels are very important for data validation in our lab. the robots actually scan the barcodes of all of the plates they're about to operate on to validate that they're operating on the correct plates. This also allows them to download more instructions, such as which wells to select liquid from for the particular protocol that they're performing. As you might imagine, when you have a whole number of very similar systems or similar records, actually finding the right record is pretty complicated. So we've exposed a search interface to our users that's powered by Elasticsearch. Updates get sent to our REST APIs, persisted on our main data stores, and then replicated out to Elasticsearch. Our administrative interface can also be used to trigger a full rebuild of the index. Elasticsearch has a capability where you can build one index while serving a different one, and then swap over to the new index atomically, which we find very useful. Another useful feature of Elasticsearch is its capability to do dynamic mapping. With dynamic mapping, you don't provide it a specific list of attributes that it's going to search over. As you post new records to Elasticsearch in the form of JSON objects, as it sees new attributes that it has not yet discovered, it simply adds those to the search index. So as our users add extended attributes to their data model, they're automatically enqueued for searching. Let's move again forward into the test phase of our system. Result analysis happens in a number of ways, some of which are automated and others are manual. This is a screenshot from our LIMS front end showing a manual analysis of fragment size. So DNA fragment length can be visualized here as a heat map of concentrations within each well. When trying to build DNA, we have a target length that we know that we are expecting to build through a set of concatenations. So we can quickly see at a glance that on the left side, we've successfully constructed a number of items that are approximately 200 bases long. On the right side of this plate, we see that the results are much more of a mixed bag. So perhaps something went wrong with those wells that we need to investigate. There's also a number of other quality control concerns with other sequence information. Sanger sequencing is one way that we can read the specific bases at each location in a genome. Sanger sequencing provides you with peaks representing the confidence or probability that a particular nucleotide is present at each location. So how do we know if we've got the DNA sequence that we hope for? A linear cutoff model is not going to cut it. So instead, we've trained a decision tree classifier that allows us to judge whether or not this is a good sequence or a bad sequence. This feeds into our quality control system. Quality control scores are put next to each sample in our system in the context of all of the other information in our front end. This includes a pass or a fail score, as well as a numeric score that can rank passes or fails more subjectively within that binary distinction. We also store a bunch of other metadata, such as the parameters for the analysis and the reason for each particular decision in case we want to know the root cause of why something failed. We also provide our users with buttons that allow them to easily change these values so that they can correct a running workflow quickly or so that they can help improve the data set for the training of future machine learning models. Let's finally talk about the analysis phase. In the analysis phase, we have to integrate data from a number of different sources across our entire stack. This includes the genomic data that I talked about earlier, as well as image data that we visualize with image processing algorithms to measure colony growth, which is a measure of health of the organisms. We also have several other measurement data sources, metadata about tracking the plates through the factory, operational logs for the robots, environmental sensors, and so forth. And all of this gets boiled down into our performance improvement analysis, understanding how much did the performance improve and what are the error bars around these measurements. This graph shows a number of different genomes and their performance. And as you can see, most of them have a strong central tendency. There's no benefit from the genomic changes that we've made. Some strains, however, show marked increases in performance. So we'll probably aggregate these changes together for our next cycle of engineering. Still, other changes didn't work out so well. Now, on the surface, these are disappointing, but they also offer us clues as to locations that are indicated in the metabolism to the chemical we care about. But we didn't make the right change there, so maybe we'll do different changes in those same locations next time. So to wrap things up and summarize, automated systems and the Internet of Things approach are changing how we approach biology and what we can accomplish. Big data plus machine learning plus automation provide us with efficiency and flexibility in our systems. On the lower, more tactical level, some lessons to take away from this are that your data model needs to be as or more flexible than the underlying process. This sounds kind of like a banal observation, but it has deep ramifications for your design. We've turned to Avro for flexible data modeling and Elasticsearch for data discoverability. But we also found it crucial to make sure that our users could extend the data model without help from the engineering team by, say, manually extending an Avro structure. Finally, we found it important to allow our users to override data by hand in a lightweight fashion. Clean up as you work. It helps the downstream analysis happen much faster. Once again, my name is Aaron Kimball. Thank you for listening.